Well, hello there. Welcome to the place to be, party people. We're gonna wrap this sucker up. So, last time we said that there were two categories of tests for coronavirus. And the first one was to see if you have coronavirus right now. And that's important so you can know if you're going to spread it to someone else. And the way you do that test is by something called reverse transcriptase, PCR, which essentially just means that you get some of the viral RNA, if it's there, and you make a buttload of copies of it, and enough so that you can detect it. And that's how that works in a nutshell. I know that explanation was pretty confusing last time. Don't even worry about it. The next one we're going to look at is this other kind of test, which is to determine if you have had coronavirus. And this is really important because this will tell you if you can go back to school, go back to work, go hang out with your grandma, whatever. Because if you have had it and you have recovered, then the thought is that you are no longer able to pass it on to anybody else, which means life can go on. So these tests, these ELISA tests, need to get going on a big scale ASAP so we can wrap up this shelter in place stuff and get on with our lives. So that's why these ELISA tests are important. So that's what we're talking about today, is these ELISA tests. I'm not going to do it with this PowerPoint. I'm actually going to do it with old-fashioned paper and pencil. So let's check it out. Sorry, let me just go to this slide we're going to need later real quick. Uh-oh. So sorry, but if we were in class, I'd be floundering too, just like this, so no big deal, huh? Man. All right, I'm going to come back. Oh, it's working. Oh, it's working. We're going to need this slide later on. Step one, don't worry. Okay, but we don't need it right now. Right now, we need this document camera right here. Booyah. Okay, Tookie friends. So, Friday, April 17th. We're going to learn about ELISA. And ELISA is a cool sounding acronym for an assay or a test to see if something's there. And the assay is called the enzyme linked immunosorbent assay. And we don't even care why it's called that. Sometimes we care, sometimes we don't. This time we don't care. In the news, they call this test a serological test or an antibody test. So when you hear them talking about serological and antibody tests, they're talking about ELISA. Another example of an ELISA test is a pregnancy test, where you pee on the device and it turns a certain color if you're pregnant and a certain another color if you're not pregnant. So these tests are pretty common. So once again, this test is determined to determine if you have had coronavirus and recovered for it. Super useful. In order to make the test happen, antibodies are used. And our bodies make these antibodies all by themselves, but we can trick slave cells like hamster ovary cells and stuff in the lab in order to make these antibodies for us. So either way, these antibodies are proteins that our bodies make naturally, and we can also trick cells into making them for us. So here's what they look like. This is a protein right here. This is another protein right here. So an antibody is actually a protein complex. And for every type of antibody in your body, this part is the same. There's thousands of different kinds of antibodies. And what makes them different is not this part. What makes them different is this part right here. This is the business end of the antibody. And in every kind of antibody, this part is different. For every kind of antibody. Okay, so let's look at some antibodies, or some models of antibodies, I guess. Which of these antibodies would stick to this antigen? So let me tell you what an antigen is, and I need to go back to this slide now in order to do that. Let's see if I can get there. Booyah, right here. So here's this picture of coronavirus. And you'll remember that all these things are proteins except for this red stuff, which is like a phospholipid bilayer, and the squiggle dog, which is RNA. But all these other things are proteins. And the spike protein over here is the one that, that your body uses to import the virus inside of its cells. So each one of these proteins can be considered an antigen, and all antigen means is that it's a foreign particle that doesn't belong in your body that your immune system recognizes as a foreign particle. So dust and pollen and things that cause allergic responses can be antigens, and also proteins from viruses and fungi and bacteria can be antigens. So an antigen is a particle that's in your body that's not supposed to be there. So coronavirus has all these different parts, all of which are antigens. So this spike protein right here is the antigen that we are going to be interested in here. So here's that spike protein right here off of one of those coronaviruses. So which of these antibodies would stick to this antigen? What antibodies? These antibodies right here. Trip out on them and then tell me which one of these 4 antibodies would stick to this antigen right here. Would it be this one, V-shaped business end? Would it be this one, half box shaped antigen? Would it be this one? Would it be this one? Yeah, that's right. It would totally be this one. This antibody would stick to this antigen. And that's how your cells can tell other cells this is the bad guy. They use these things called antibodies as tags to tag the bad guy as being a bad guy. Sounds good? So hopefully you kind of understand what antibodies are. Now, how can we as biotechnicians use antibodies to get ELISA done? And we're going to use ELISA to tell if a patient has coronavirus or not. So it starts with a plastic test tube, and just serendipitously, plastic binds proteins really well. Plastic and proteins have electrical properties that are complementary to each other, and proteins stick to them. So you can take a plastic test tube and you can treat it with spike protein from coronavirus. So you can just get a bunch of coronavirus in the lab, chop it up, separate the spike proteins from the other proteins with chromatography, put it into the test tube, wash it around, dump it out, and the spike proteins are going to stick to the plastic. They'd also stick to the walls over here also, but I just wanted to keep it sort of not cluttered. So here we are with our plastic test tube with coronavirus spike proteins. This coronavirus did not come from a coronavirus patient. This coronavirus came from the lab where it was manufactured. So we can, to this test tube, we can now add blood serum from a patient. So blood serum is the blood, all the parts of the blood that are not cells. So you just take the blood, spin it down. Actually, you take it from, take the blood, spin it down, and get rid of all the cells, and then add the serum to this tube right here. So here we are adding that serum to the tube. And in that serum, there's all kinds of stuff. There's platelets in there, which we can symbolize like this. And there's random antibodies in there, which we can symbolize like this. And if the patient has had coronavirus, then they're going to have antibodies that stick to this spike protein right here. And the reason why is because that patient will have had an immune response to the virus. And in that immune response, they will have produced bazillions of antibodies that bind to the spike protein and label it as a bad guy. And incidentally, that's how you cure yourself of viruses. It takes a while, though. It takes a couple of weeks. So we got all these here. I'll add some other random protein or something in there like this. So we got all this random stuff, but this antibody stuck to this antigen. This antibody stuck to this antigen. All these other proteins, including these antibodies, didn't stick to anything. So what we're going to do now is wash the tube out gently with some nice clean water, dump it out, and then what you're going to have is you're going to have the antibodies that were stuck to this antigen when the antigen is stuck to the tube, and that's going to be all that's in there. Everything else is going to get washed away. And you're going to be like, well, Mr. Gator, you said proteins are attracted to this plastic, so why didn't all that junk just stick to this plastic? And it's on account of, I didn't tell you, one step you do after you bind this is you add a bunch of milk to your test tube, because milk is a cheap source of protein, and the milk proteins just stick to all these places that these antigen proteins haven't stuck to, and it prevents any of this other stuff from sticking. Sorry, that was a little bit of an aside. So what you're going to do after you have this nice clean system right here with these anti-coronavirus antibodies is you're going to add, I like this part, an anti-antibody antibody. Oh, you're going to add an anti-antibody antibody that includes a fluorescent tag, which we're going to symbolize with this this thing right here. So we got these antibodies from the patient. And then what we're going to do is we're going to add antibodies that we manufactured in the lab, or someone manufactured them for us. You can just buy them, you know. And we're going to add those anti-antibody antibodies. What do anti-antibody antibodies stick to? That's right. They stick to antibodies. So here they are right here. Anti-antibody antibodies that include a fluorescent tag. Boom, boom, fluorescent tag right there. Then we're going to wash the tube gently to wash any of these fluorescent tagged antibodies out that are not bound to anything, that are not stuck to anything, because there's a bunch of random ones in here still that aren't stuck to anything, you know, like this. And that's going to give us a false positive if we don't get rid of those suckers. So the next thing we do is we gently wash, and then all we have left is these anti-coronavirus antibodies, which are stuck to this coronavirus antigen. And then we've got this anti-antibody antibody, which contains this fluorescent tag, and all these other ones that weren't stuck to anything are now washed off, and they're gonzo. So what we're going to do now is we're going to take our test tube, put test tube into a spectrophotometer. Oh. What do spectrophotometers measure? Light. That's right. Light of a certain frequency. And we want to measure light of this frequency, this fluorescent antibody right here. We're going to put that sucker in there to measure light. Yeah. So if you get light being emitted from the test tube, that means that your patient is positive for having had coronavirus. And if there's no light, you got it, brothers and sisters. If there's no light, that means the patient is negative for having had coronavirus. So that's how ELISA works. Probably maybe what's being done as we speak is researchers are trying to develop a pregnancy type ELISA test that can be done without a spectrophotometer. So it can be done just visually by looking to see if a reaction happens or not. And that's what's going on right now. Once those tests have been developed and manufacturers have figured out how to scale them up so you can make a buttload of them for really cheap, then they'll start sending those suckers out. And they'll start doing studies on the human population in the United States to see how many people have had coronavirus. If it's determined that enough people have had it for us to have developed herd immunity, then we can let the old folks party again. Alrighty then, that's it for the coronavirus briefing. I hope you guys enjoyed that. And I'll see you next week. TGIF, you friends. You guys are so friendly. Can't even believe it. What are you guys even doing right now?